All
I
right.
just did, Lizzie.
Oh, thanks, Katie. Sorry. I wasn't sure if you want me to do that today. All right. Well, hi, everybody. Welcome to the November installation of the Symbiota support group. Today we have Gary and Frank from the Global TCN doing a presentation, but we are going to just make a few announcements as usual before we get started with today's official topic. And so, maybe if you haven't heard yet, this is very recent. So, the Myco portal, which is the Mycology Collections portal, which was previously maintained at the University of Illinois, it was moved to the Symbiota Support Hub / University of Kansas servers late last week. And so, if you are a user of the Myco portal, you'll have to reset your password, and the new help desk is the Symbiota Support Hub help desk. So the regular one that you're probably already always used to is just help@symbiota.org. Just be aware of that because in the past it was help@mycoportal.org. So just want to clarify that for anybody who's part of the MycoPortal community. And then, of course, this portal got an upgrade to the latest Symbiota code base as part of this migration. And now we're working to set up batch image ingestion for this portal community. So stay tuned if that's a service you'd like to participate in.
One comment about the resetting of the passwords is that is going to be prompted soon. So you don't have to reset your password right now, but we have to do a configuration change that is going to make you reset your password soon. So um, if you start to log in and then it doesn't work, that means that you need to reset your password.
Got it. Thank you for clarifying, Katie. I didn't realize that. Another announcement is that in about a week, the Entomological Society of America is having their annual meeting. This year it's in Portland, Oregon. And this year, Mark Fisher from our development team will be representing us. Mark will be giving a presentation on Sunday morning. And if you were there last year, you might remember that Katie and I, or it was Samantha and I actually, at the iDig Bio booth. We won't be having a booth this year, but Mark will be there. So if you see him around, say hi. It's kind of a nice opportunity to talk with our developers who aren't always representing us at these specific meetings. Another announcement, also for our entomological collections communities here, is that the Ecdysis Steering Committee is still looking for material sample beta testers. And so here's just a screenshot of kind of like what this module looks like. It's just another way to document your collections, but specifically kind of like subsamples of your specific specimen record. So if this is of interest to you, they have some controlled vocabularies that could use some testing. So just email our help desk if you'd like this activated for your collection in the portal. And then lastly, we did send out an email about this recently, but just another reminder in case you didn't see it. Publishing to your data to GBIF is a great way to increase the visibility of your collection and create a secondary data backup. So if you're not doing this, or if you know a collection that should be but isn't, it's usually pretty easy to set up, especially if you're using a Symbiota portal as your primary database of record. We can help do that for you. Just email help@symbiota.org and we can set you down the path of making this happen. And I think that's our last announcement before I pass it off to Gary and Frank. Stop sharing my screen. Righty. Are there any other announcements I should ask before we move forward? Going once, going twice. Nope. Okay, go for it.
Okay, so welcome to our meeting, and Gary and I will talk about various different things that we developed as part of the global grant. And I will start it off with first sharing my screen. And so you all are obviously aware that the global grant covers two different portals, the lichen portal and the bryophyte portal. And since I'm a lichenologist, I will mostly talk about lichens today, but some of it actually will affect also the bryophyte portal. And so what you might have all noticed is that the map search was entirely revamped. So when you go to click on search, map search, then that those maps that are showing up are now from Leaflet, from OpenStreetMaps. And what we discussed when we started the grant quite a while ago, we wanted to have like a super portal where you would be able to search both the lichen and the bryophyte portal. And then with talking with the programmers, we decided that wasn't quite feasible and was not the way to go. But what makes things more easily to integrate, and that's something that we developed as part of the global grant is an API. So basically an application programming interface, and there's some documentation for these APIs actually available. And what I have to say is that those are still a bit experimental. So people can use them, they're functional, and actually the API is using some functionality in the portal, especially in the map search now. And so it means that different portals can now talk to one another. And the only ones that do that at the moment are the Briarfied portal and the Lichen portal. So when you do a map search, you can now in the Briarfied or also in the Lichen portal, you can enable what's called cross-portal search. So you can search not just one single collection in one portal or several collections in one portal, but you can also search several collections across two different portals. In this case, only the Briarfied herbaria from the Lichen portal or the Lichen herbaria from the Briarfied portal. So when I do this, for example, I would look for Buellia spuria as an example. And I would like to know which Briarfieds, for example, Burium argenteum, might co-occur in the portal, the records of both Buellia spuria from the Lichen portal and then Burium argenteum from the Briarfied portal. And if I do a search like this, it will take forever because it's actually two of the most common species in both portals, and then it will show all the records from all the world. So you get a map that looks a bit like this. So you see there's from the Briarfieds and from the Lichens are these different records. And what currently, the way this currently works, and this is a little bit unfortunate, I personally think, it doesn't query the taxonomic thesaurus, so you get all the synonyms split up separately. But to move this a bit further and do this a bit quicker, you can also restrict your search to, say, for example, the state of California. And then if you click on this, it'll take still a while to find all these records. And while I'm talking, the little spinning wheel is running. And then basically it only shows you Buellia spuria and Burium argenteum from California. There you go. And of course, some of those records might be having wrong locality data, and then they show up too. But here you now see how these two species co-occur together. In the taxon list, they are color-coded. So there you got your Buellia spuria, and you can change these colors. You could say, okay, this one I want to have, say, red. And now they're standing out more. So those red dots are the Buellia spuria records. And you could say this one, the yellow one, I also make green, different kinds of green. And that way you can more easily see how things co-occur, grow together. And so that's a big advantage now, that basically whenever a new Symbiota portal joins and is installed somewhere, then the API will automatically connect these portals. And in the future, theoretically, you could do searches like what kind of tardigrades can be found in association with what kind of Briarfieds, or which bees are found together with which vascular plant species. And that means that you can do these map searches now across different portals. And in the future, hopefully, you can do all other kind of more complex searches across different portals. And for people who are technically inclined, they can actually look at those Symbiota documents. So they all have the same sort of thing. They use the portal and then the API and the documentation. And the documentation explains in detail how the API actually works. And this is still like a little bit in beta, so it's not entirely finding everything in all fields of all the Symbiota portals, but it works fairly well. So then the next thing I wanted to talk about is that in the Lichen portal, when we started this exercise, we decided that the help and resources were, at the time, that was before the global grant, quite inadequate. And so at the time, I decided I would launch a little WordPress site, and that WordPress site then had a lot of tutorials how to do data entry, how to use the portals. This is largely superseded now, of course, by the help of the Symbiota team. And so instead of doing this classical search support for help, we decided we kind of revamp that site a little bit, and we create a resource page with various different might be used by people not only for liking but for various different other portals as well. So when you go to this help and resources menu here, you see the classic Symbiota help there. You see an item that I will talk about a little later, the glossary, but you also see this thing called consortium resources. And that consortium resources then takes you to a separate page. And I keep putting news on what new things and new developments we have. And that help and resources page is still having those old tutorials. So for example, a beginner's guide, how to do data entry, a guide about checklists, and there's one for simple checklist. This is the old one here, and there's a more advanced one, and Gary will
Frank,
talk about.
I don't want to interrupt you, but your volume went down a little bit. Maybe try getting a little closer to your mic. It seemed like you maybe leaned back just a touch, and we could still hear you. It was just quieter.
Okay, tell me if it, I mean, because I had some issues with the. Can you hear me now?
That's much clearer. Thank you.
Okay, so I just put the little thing in front of me. Anyway, so now I need to check my outline where I was going to talk about. So okay, one thing that I recently launched is when you deal with various different groups of organisms, often enough, specific groups like lichens require certain techniques how to collect them and how to maintain them in your collection. And so I noticed that a lot of people then, when they collected things, were doing it in very different ways, and the same with curating of specimens. So I recently launched a tutorial on collecting specimens and another one on how to curate material. So these are very detailed. Why would you collect specimens? What you want to collect? What kind of material would you collect? So that if you have a lichen round like this, you would at least get a chunk that has both the center of the thallus and the margin of the thallus, because as you can see here, fruiting bodies in the center are representative as well as those low margins. And so there's a lot of information that you might want to collect and how to do it more efficiently. And one thing that, of course, and I talked about this in Palm Springs at the botany conference, when you collect, quite often people are kind of lazy and sloppy about what kind of habitat information they collect. So the way I kind of tend to make this a little bit more efficient is I have a collection book where to put the major locality site. Then my collection bags are simply labeled by these locality acronyms and maybe the date. But then I also have a little piece of paper that I stick into these collection bags that are basically having a little summary of quick taking notes of habitat information. And here, for example, I can say, okay, I collected this at this locality site in January 2019, and it was growing in a habitat below large saguaro on rock. And the rock I collected it was rhyolite, semi-shaded, wind and rain exposed, the front of the rock, and it was a big boulder. And the whole thing was north exposed, and I even took a photo. So by standardizing this information, then of course you can also, once you got this into the portal, you can then get the information out, and you can statistically analyze the stuff. So here you can see that these are a couple of species from Galapagos, and this Arthodium galapagos, for example, grows typically at the front of boulders, whereas this one here is more associated with overhangs. There's other species that are on the top, in some cavities, etc., etc. So you can basically come up with a species habitat profile using that information. And in the portal, what many people are not necessarily aware of either is there are actually now all these habitat data that can be coded into the collection. So when you go out and you come back from the field and you enter your data and you go to your specimen database, then there's now options in the consortium, in the lichen consortium, to add habitat data. So they're called traits. And so you can say, okay, the trait wind rain exposure, something is wind and rain exposed, right? Or shelter. It grows in a sheltered habitat, or it might be in a sunny or semi or shaded habitat, north facing. And so you can code these traits. But another way of doing that, instead of doing this manually for each specimen, is you can also, from existing collections, harvest this data. So when I did work in the Galapagos, I need to go to the correct one. Sorry. So I worked at the Charles Darwin Foundation in the Galapagos, and they have their collection data in there. So this is CDS lichens, the Charles Darwin Research Station. And now what you can do, you can select a particular trait. For example, you can say substrate. And then you might want to go in the substrate or in any of these other fields. You can say, please look for anything that says, for example, and I think I do have that mostly in occurrence records, trunk, and then get my field values. And now I've got all the data that has trunk in occurrence records. And so now I can check and say, okay, please code me everything that has trunk, 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 and code it as growing on bark or lignicness, etc., etc. And then maybe you want to put expert needed or not. And so you can batch assign these different habitat traits automatically to your specimens. And when you then harvest that data, you can then use something like PCR here to analyze that data. So that's also something that we developed and implemented as part of the global grant. And I still want to publish on this because for the Galapagos, you get some really nice habitat profiles to analyze your records. Okay, then the support side here has a lot more to offer. And among those, we also have some programs that help you. For example, the global emphasis was much on digitizing specimen. So one little program that we developed is called the BCR Watcher. Here's the site for the BCR Watcher that you can download, and it has very detailed documentation. And just an idea on how the BCR Watcher actually works. BCR means actually barcode renamer. So basically, it's a little program that scans barcodes in photographs and renames the files according to the barcodes that it finds in those files. But it can be much more powerful. So this is what the program looks like. So basically, when you configure it, you say, okay, I have a watched folder on my computer. So when I take a photo, I put the program into watch mode, and then it will scan watched folder for images. And it will detect images in that folder with a barcode, and then it will rename those files according to the barcode that it found, and place this file that it found after renaming it into a destination folder. It can be anywhere on your hard drive. It can be actually on different hard drives, depending. All you need to do is you need to make sure that that you configured correctly what kind of code page. And currently, the barcodes that that support is either 39 or 128. And in these instructions here, so there's some gibberish about how to recognize and distinguish different barcodes that the program currently is able to read. And then you also need to put letters that are typically the prefixes of your collection. In our case, it's ASUL, and then the number of digits that are included in your barcode. Now, what the program can do while you're taking information, it can do three different things. It can just rename the files, and that means that each file needs to have a barcode, and then it will just simply rename the files, and the files will stay in the watch folder. It can also prompt you for each image, and it will give you a data entry mask where you can enter data. You can also configure to take photos by specimen, meaning that quite often you have a scenario by which you take photo of a label first, and then you take another photo of that same specimen, but actually the specimen itself, and maybe then you take another photo of a section in the microscope. And this by specimen mode means that it will basically assign the barcode according to the first photo that it takes to all the following photos until this turn off the watch mode again. And the last mode is rename and move. It's an assumption that each photo will have its own barcode, and after renaming it, it will move it into the destination folder. And it works with vascular plants, so large sheets, with lichens, or with bryophyte packets. And now I mentioned already that the API works not only with the map tools, but you can actually update the database, the names that the program stores, from 3 different portals, from SciNet, from the lichen portal, and from the bryophyte portal, and it can also update the list of XCCAR-T. So just to show you how this is supposed to work, that the program reads the barcode, it adds a suffix to your barcode, and then it places it in the destination folder if you have it by specimen. And I currently have a couple of images in the watch folder. So if I click this to watch, then what the program will also do is it will generate a separate sidecar file, a CSV, with all the information that you put in, the metadata, but it will embed also that same metadata into the header of the photos themselves as so-called XMP. So you can click on this, and here now it says it has images found. So normally you would have your camera running, you take a photo, you have the program in watch mode, and the photo will also automatically pop up. And now you can add all this metadata in here for that particular photo. And then when you click continue, that photo will have the metadata embedded, and it will also create a CSV file of skeletal metadata. So say, for example, you have a scenario where most of your collections are not yet databased. And instead of manually having to enter it online into the portal, you can create these skeletal records that you can then upload. And once you have those records uploaded and you then upload the images, they are automatically being associated with the skeletal records. So I will now abort this, but this information that you put in, like the barcode, the collector number, and so forth, you can configure which kind of metadata you want to put in. So you can select a bunch of fields that you can record while you're taking photographs. And those fields then will be written out into the CSV, but they will also be written to the image files themselves. And then if you use a program like Lightroom, that program will be able to read that metadata, and you can use it for databasing your photographs. Okay, so then I mentioned that we, as part of the global grant, also were working on a glossary. And so again, if you go to the help and resources site here, you click on glossary, then you can look for terms that are associated with particular groups, or you can look for all terms, and you find image definitions of terms by simply typing in that. So for example, if I put Lichen Orion in here and I search, you will see there's a long list of terms. So you can narrow this down by searching and browsing for those terms. And here you have the definition for crypto Lichen Orion, Lichen Orion, Lichen Orion. And this is the pop-up that comes up when you want to look at what is the meaning of Lichen Orion. There's actually a Spanish translation of this. And there's a couple of images. Those, unfortunately, they don't pop up right now, so you can't click on them. But what you can do a right click and then show them in a new tab. And then you can actually, where's the new tab? It's back here, then you can see those images at larger magnification. So the glossary, I'm kind of losing it with all the tabs now, has a lot of terminology. And the nice thing is when a terminology pops up, there's also other terms that are underlined, and you can go back and forth between those different terms, and you can read about them and learn about them. And lots of them, they have images like these. Some are linked to specimens. So for example, if you go and search for umbilicate, then there's actually a photograph from Bruce McCune. And if you click on that link, then you will see the specimen information of that particular specimen from which that image was taken. What you will also notice when you search for all the terms, that the program has not only terminology like things are absent, present, accommodate, what does that mean? But it also has a lot of chemistry information. So for example, if you look for atranorin, which is one of the most common cortical substances that found in lichens, then there's a definition of atranorin, which is a little bit different from the general definitions. But basically what it gives you all, if you're familiar with thin layer chromatography. So here are the different solvents. So for example, the RF value of atranorin in solvent C is 79. So it gives you all that information from the atlas of Jack Helix in Australia about the secondary metabolites. And it also shows you what the secondary metabolites look like when you run the TLC plates. Nicer than that even is that we, again, if you go to help and resources, to the consortium resources, you can actually download a program called WinTabalites or MyTabalites, and that software, MyTabalites, for the analysis of secondary metabolites, connects again through the API with the portal. This is what that program looks like. So for example, you can say, I add a TLC spot now at 35 and 10. So these are the different solvents. Those are the spots that are on the plate, and then the program queries the database and it returns you possible secondary metabolites that match these criteria for that particular spot test, for that spot on your TLC plate. And there's a huge dictionary of substances online, and you can update the substance data from the lichen portal. You can manually add substances, which is not recommended because the next update you run, those entries will be overwritten. But you can also view and edit the substance records. So for example, if you look at atranorin, then the same information that you get in the glossary is here displayed in a much more dynamic way. So for example, you can see and have a look at these images of the chromatograms. And you can, when they're available, you can also compare substances. So atranorin is closely related to chloroatranorin. So if you want to compare these substances, you do a right click and then they're being presented side by side in different windows. And you can see that atranorin is found at 79 and chloroatranorin is a little higher on the plate. And you can actually say, okay, where were these things found? So for example, this one is found in Alectoria sarmentosa. So now it pulls from the lichen portal an image of Alectoria sarmentosa together with all the other substances that are found in Alectoria sarmentosa. And you can copy these substances easily, and you can put them into, for example, so there's the substances, the list of substances that occur. And that can be very, very useful when you want to know, okay, is it possible that my specimen is Alectoria sarmentosa? Are these substances found in Alectoria sarmentosa? And of course, instead of browsing the list this way, you can also go to view a list of lichen names, and you can put any name in there. And then the program pulls from the lichen portal that information, what substance have you found and reported from that species, and adds a little photo, or you can go right directly to the taxon profile of that species in the portal. So I think from when I'm looking at my list, I'm mostly done with my part of this now. And so the next section of our presentation today will be by Gary Perlmutter.
Yes, hi folks.
But I'm not
So,
sure whether we should take a little break in between, and just if people have questions. I don't know.
I think it would be a good idea just to have a little pause.
Because it might
sure.
be a bit overwhelming, all the stuff that are presented.
Yeah,
Sorry,
good
Gary, I didn't
chance.
want to cut you off.
Do we have right off the bat, are there any questions from anybody in the audience? We'll probably have a few more minutes at the end too. Okay, I don't
Everybody's
think.
happy. Nobody has questions, so that's great.
Oh, Chilla says the map is just points, not polygons, right?
Yes, the map is only points at the moment. Yes.
My other question, which I think I've asked in the past related to BCR Watcher, that's still just on PC. There are no plans to convert that to like a Mac-based program.
Actually, I have managed to get it run on Linux. There's a software called Wine, and it runs really very well on Linux. And I need to play on a Mac to get it installed there. Actually, I tried getting it to run on, and I managed to get it to run on Mac. But Mac is a bit more complicated. You have to first install, there's actually instructions that I can show you. So if anybody wants to try this out, you're welcome. So you go to, again, your consortium resources, and there's a little thing called running. I hope everybody can see that. My tablets on Linux and Mac. And so Linux is fairly easy. And Mac, you need to install something called Handbrake before you can install Wine that runs also on a Mac, because Linux and Mac, they're kind of similar. And so theoretically, you should be able to run it on Mac OS. And I'm actually, since Manuela has a Mac, I'll plan to try to install it this afternoon. And then once I got it up and running, then I put a tutorial on how that works.
Awesome. I didn't realize that was possible.
Yeah, there's some simple programs that are not too complicated that you can run Windows programs on Mac through that installation thing called Wine. I don't know what that stands for, but
All right. Well, Gary, would you like
Okay.
to transition to your segment?
Sure, sure. So my presentation is a little more focused. Basically, I'm just going to be presenting on some of the advanced features in checklists, because I've been recently working on a checklist directly from scratch using a lot of Frank's innovations that he's put together for the Ecuador checklist. So let me go ahead and share my screen and see where we can start. So one moment. Okay, so can you see this okay? So this is the checklist that we got going on. This is in its draft version. And basically this is a checklist for the South Carolina state. And like I said, just started this about maybe just a month ago. And following Frank's lead with Ecuador's, one thing we want to have is all of the synonyms listed, because we've got a number of specimens that were originally described from older names. For instance, like a Ravenel here. We also have vouchers, and if you click onto the voucher, you're going to get the specimen name, record itself. And here you got the original name, Abrothalus smithii, whereas the current name is Abrothalus parmeliarum. And we also have some notes in here, such as the type of growth form it has, and where it grows on, and some rough habitat data, and also some cautionary notes, such as whether the species is truly within the state, or if we need to look at specimens that are that have been cited. And also we do have some citations as well, such as here. One of the biggest citations is Alex Siegler, who put out a preliminary checklist back in 1997. Needless to say, because the taxonomy has moved on so much since that time, a lot of his specimens, while the records are temporarily flagged here, they do need to be re-examined just in case. So with that, let's go ahead back to the home page, and I'm just going to go over here to another page. Nope. Here we go. And we go home to home page. So what I'd like to do is at the consortium resources, there is a link here for checklist itself. And here we got two parts. We got one, how to create checklists. And that's pretty much how I started the checklist, myself. And if you go into here, you do have a bunch of instructions on how to create a checklist, such as a little green sign to create a new checklist. And actually, we can go ahead and do that. So basically what you do is you go into back to the home page, and you want to go into your profile page. And in the profile page, you got like 3 tabs here. The first one is with the checklist, and just click on this right here. And this is how you create a checklist directly from scratch. So you just give it a name, put in the authors, and insert any information from abstracts. Sometimes you have a published checklist in the literature. You can just copy and paste that abstract and put it in here. For the South Carolina checklist, let me get back to that. Okay, I got too many pages open here, so just excuse me for a moment. Just going to go back to the profile and to the South Carolina checklist here. If you look here, you will see that, no, this is not the one. Excuse me. Let me back up a little bit. And okay, so for the South Carolina, this one right here, you're going to see a little click for more detail. In here, I've got just, the citations where the authors are from, and also a note that when this was last updated. And also we got these 3 little toggle switch here, which will help you manage the checklist after you built it from scratch. So going back over here, you're going to see this very same thing once the checklist is created. But before you're doing so, what you can do is you have a number of options here. And you can put in your lat longs, you can put in a fingerprint or a polygon, and also, then you want to put in what view setting you want, whether you want to have with private, which is what I have currently because it's not quite ready for publication, or you can have strictly private with only editors can view, or you can do it as public too. So, and then after that, you just click the create checklist, and then you have the checklist itself showing up. So, and let me just close this a little bit so I can see my tabs. All right, so going back to the checklist itself. One thing it does that's really cool is that once you've created the backbone of the checklist, you want to find out whether you can add child checklists to it or not. As you can see right here, we got a collection of checklists that were here. And the first step I did toward creating the South Carolina checklist is to add these individual checklists, pre-existing checklists, into this parent checklist. So, and the way I did that was by going up here into species checklist. No. Okay, so going into this section right here, going to checklist administration. And here you got a series of tabs, and you go into the description, and in the description is just like that blank form that you saw before. But also you have another tab called related checklist, and this is where you get your child checklist. So basically what you want to do is if you want to add another checklist, you click the click box, and then you have a pull down menu over here, and you can just select any one of these. And once you do that, you click add child checklist, and all of the vouchers and all of the taxa will be transferred onto the parent checklist. And that's how it grows. So that's one way of growing the checklist. Another way of growing the checklist is by selecting a number of specimen vouchers. This, from the consortium itself. This is really recommended for those collections that you trust or whose identifiers that you're most familiar with. So, one thing I did is that for the South Carolina checklist, the first thing was to just to batch load all of the specimens from my home institution, which is NCU. So basically how I did that was I would go back to the checklist itself, go into the vouchers, and here you got a little pencil icon here, and this gives you a set of parameters or fields from which you can zero down your search field. So basically, if we want to search instead of all collections, we just want to check all of those. Let's see, from Brigham Young University, and then we can keep that as a safe search term, and then we see how many specimens from Brigham Young University you can see that were collected in South Carolina. And this is the number of taxa that were from that area, from that institution that don't have vouchers. And if you want to link a voucher to it, you can just click this button here. But it would be better just to look at the vouchers and see what's available. So this goes to the actual so this one goes to the species page and gives you an idea of what that is. If you want to click on the voucher link, this gives you a list of collection vouchers that are in various institutions that you can review to, whether you want to add them to your checklist or not. And basically, as you can see, you got a number of historical specimens, some from Ted Ati. Those are definitely reputable. And then if you want to add those, all you have to do is just click this button right here, and it'll add that particular specimen over to the checklist. However, if you're comfortable with everything that's in here, you can then go to this display mode and go from here. You can go into occurrences for non-voucher checklists, and see there aren't any there. And so actually would be, so those are all the non-voucher checklists. And then if you go into the missing taxa, this is what we'll get into. You go into species list, you can see they got 3 species that are from the search, but not in the checklist. And if you want, you can do the batch linking if you're comfortable with them. And here it gives you a list of specimens with those species, and you can look at them more closely, see if this looks, reputable, and if you want to think it's worthy to be within the checklist, if it's verifiable, and then you just click on each one of these. Or if you're comfortable with all of them, then you just click on this one, and it'll add those, and then you just click some vouchers, and they'll automatically go on to the checklist. So going on from there, there are some other tools that you can use. So if we're going back to the checklist, as I mentioned, there is a number of notes that you can add to each species. So how you do that is you want to click onto the species icon, and it turns on once you click onto that. And when it's off, you don't see any icons, except for maybe just a little related specimens view here. But if you turn on the species icon, then you have a little pencil mark and a little link for vouchers. So this gives you the option to, one, you can add additional vouchers to your taxon through review, or you can add notes to the taxon itself. And this is where you can add, whether it is a Nicholas fungi or lichen. You can give an abundance rating, any internal notes like whether it was a holotype, or if it is considered rare per IUCN categories. Over here we got editor notes. Those are not visible in the checklist. Those are just internal notes for if you are collaborating with another colleague within a given checklist. And also the source. Now the source is really important because you want to, source your citations, or, and you can just, put one here, say Siegel in 1997. And another thing that's really important is that while you're doing this and you're adding literature citations, you want to have a separate document that lists all of those citations. And once that is complete, that can be converted into a PDF. And then the PDF, you can send, attach a link to the summary page for the checklist itself. So like if you want, and a really good example of that is the Ecuador checklist that Frank has put together. So as I said, that is pretty much it. Like I said, I'm still running in, developing this. And one of the things that I've done that is a little more labor intensive is being able to go through specimens one by one, or specimen records. And how I do that is basically I would go to the search. I would, click all collections, type in my state, South Carolina. Click display because I want to see everything that's collected there. As you can see, we've got a whopping nearly 20,000 records, from the state alone. And so one way to organize this a little bit is to click onto this display sort, and you can sort them by scientific name, which is what I've done. And then you sort the results, and you go from undetermined through Abrothalis and moving on. And what I've been doing is that I would review these one by one. I would not consider specimen records that are to genus only, but those that are to species. There were, like I said, there was a number of collections by Alex Siegler that were collected back in the 90s. Only if they are supported by his checklist will I add it to the checklist. Otherwise, I'll just flag them and ask folks over at Clemson University to send some specimens that we can look at and just to verify their identifications. And basically, I've been going through all this, and over the past few weeks, gone about maybe through 10 pages of this. So luckily it's not as daunting as it could be, because there is a lot of mental weeding that you can go through. Like I said, and also to address synonyms, I also include species that have older names to them. And the cool thing for that is that once you get back into the checklist itself, one of the things you can do is to have the names converted from the original name over to the current name. And how we do that is by going, I think it's in the vouchers section. So we go into, not missing that, but non-voucher tax, no, that wouldn't be it. So let me check my review. I'm just going to check my
So there's a different display mode. If you go to the checklist itself, you can switch between original and central.
Exactly. And what I meant to do is go back into this pencil icon. And what I didn't mention, I just talked about the stuff on this box here, but this down box over here, what you can do is that if you have an older name, you can just type in the target taxon of the current name and then just click rename and transfer, and all of those records of the older name will be automatically sent to the newer name. And that is a good way of streamlining your checklist so that the total number looks a little more realistic than it would be. So to give an example, as you can see here with the original checklist and the taxonomic filter, we've got over 500 species, but if we use the central thesaurus, it's down to 488. And that's because I've done a lot of these transfers themselves. However, if I go to the original list, like this one here, and go to the species list, we have a total of nearly 2,000. So that's clearly not, the true checklist size, because a lot of these taxa, not only do we have just genus level taxa, but we also have a lot of species that are actually synonyms of the current name, or in other words, older names of a given taxon. So that's pretty much what I've got going on here. Most recently, I was given a couple of spreadsheets from a recent foray. I've gone through all of those. And one thing I've done is adding those to the checklist. And I've also made a note saying that they're from this foray. So let me just go ahead and find one for as an example. And it should be fairly, let's see here. Okay, so I'm looking here because I think there's one within Panariaceae, Parmeliella. Oh yes, Tryptophylla. So as you can see here, this one doesn't have any checklists, any vouchers listed, but we do have a citation saying that this is from that recent foray. So these spreadsheets, which have the raw data, a lot of these specimens haven't been verified yet. And actually, while some of them have been verified, they haven't been added to the lichen portal. And once they have been added to the lichen portal, I can go ahead and insert them as vouchers. So that's pretty much what I've got. Does anyone have any questions?
Thank you, Gary. That was a very in-depth tour of what you've been working on, and
yeah,
I learned.
Like I said, it's the advanced checklists.
Yeah, no, that's great.
Yeah. All thanks goes to Frank. Like I said, this is one in progress, but if you want to see a really nice example of a checklist that has all of the bells and whistles on it and all the details, I highly recommend looking at Frank's Lichens of Ecuador. In addition to that, he also has a published version of this too that came out. Great. So yeah, thank you, everyone. Thank you, Frank, for the inspiration. That's pretty much what I've gotten in building this. And needless to say, in my individual specimen record review, I'm still in the A's, and luckily this project I've got is for at least a year. It'll be extended, and so got plenty of time to add to this. And we envision the checklist total to be maybe roughly about 1,000 species. North Carolina, by comparison, has 1,500 species documented, so looking pretty good. But it's definitely higher than what Alex Siegler had reported back in '97, which was about 377 species for the state. So it's pretty interesting to see how well these checklists have evolved over time.
And just to mention that there is I taught a workshop on how to do all this. I mean, obviously everything that we've shown today is a bit rough and quick and dirty. So there will be a video, I think, on the Symbiota support hut where you can go back to all the things that we talked about. But also, if you go to the help and resources side of the Lichen portal, there are 2 tutorials, as Gary mentioned. There's one on building checklists generally with like simple tools, but there's also a more advanced checklist one. So that also explains how Gary put in these colors. So these tags for some simple HTML, so that things appear red or blue or whatever color, and some recommendation and suggestions how people might use these things. So
right. And also I've got an outline to today's presentation on the work. So if anyone's interested in that, I can certainly send it to you via I'll convert this into a PDF and send it to you folks if you're interested. Great. And with that, I'm going to go ahead and stop sharing.
I don't see any questions in the chat, so I'm going to take that to mean there aren't many questions. People probably have a lot to think about because we've covered your help and resources page, how you're all using traits and to record habitat information, BCR Watcher, glossary, and of course the deep dive into checklists. So this was a lot to think about in the Symbiota support group, but excellent. And it's been really great to see how you all have leveraged your portal to really the maximum. It's pretty impressive. And for those of you who missed Matt's presentation on weedic bios, a lot of this is part of the global TCN, and they've really done a lot to make Symbiota do a lot of the heavy lifting for them. And it's been a pleasure to work with you all and see how this has shaped up. So thank you very
It's a
much
great
for
tool.
presenting today.
Yeah, so I'm surprised there's no questions because I thought it was all chaotic.
People
I
might
was
be
afraid
thinking
we'd
about
run
it.
out of time. So I'm glad that worked out.
There's a question for Gary, I think. Gary, you mentioned how you move synonyms around in a checklist.
Mhm. Oh, right.
Is this actually
Is it
adding the
actually editing the
thesaurus?
thesaurus? No, that's the good thing is that when you move and transfer a taxon name to the current name, it doesn't affect the original record at all. It's just done, via name movement. And if you click onto a given, taxon voucher link, it'll go to that original record and it'll show the name that is listed under. Like I shown with that Avathalis parmeliarum, the link, shows Avathalis, Tillii, the original name.
So it's not changing names on anyone's in anyone's
No,
collection.
no,
No.
no, no. It doesn't touch that at all.
So basically,
Yeah, it's just
to,
in the checklist itself.
to explain that a little further is each of the bottles have a taxonomic thesaurus that keeps track of the currently accepted name and their synonyms. And so what happens a lot, though, when people put these checklists together is they put specimens under the old name, the one that's currently not accepted. And then you have these display options that you can display the checklist either in the way it was originally compiled, so in the original way how they were put together, or according to the central thesaurus. And there's sometimes discrepancies between both because obviously the way things have been identified and the way things are named now might be different. And so in order to clean that up, to clean the original checklist up and use the central thesaurus, instead of having to constantly switch between the two views, you can use that tool that Gary mentioned where you can basically remap the old name to its new name, and thereby you're eliminating the old name from the checklist, but you're keeping that link to the voucher specimen, to the specimen that you cite, still intact. Yeah.
Right. And
The
also in
other,
the checklist itself, if you see a name, you have the option to display all the synonyms on it too.
yes.
So you can see all the synonyms as well as any vouchers that you choose, whether it's under the current name or a synonym. It'll still show up
Yeah.
under that one name, if you choose the
So
central thesaurus.
the other option is what Gary showed is that he went to the search and he searched for everything from North Carolina. But you can actually do that straight from the checklist. And as he has shown with that voucher tool, when you add vouchers to the checklist, there's a little check box at the very bottom of adding those specimen names. And you can add them according to the current taxonomy that the portal currently maintains. And then, even though the voucher is identified under a different name, it will be added under the name that the thesaurus currently accepts. And that helps with preventing to have a lot of cleaning up to do later on because it will add the names according to the current taxonomy of the taxonomic thesaurus. Now, there might still be things down the line, though, because names keep changing. So if you look at the Ecuador checklist that I put together like about a year ago or so, and you switch between these different views, according to some changes that might have happened in the meantime, you now might get again some discrepancy, and then you can go back there and use this remapping tool to not change the identification of the specimens, but simply mapping the old identification to the new ones. And so there's various different ways to achieve the same thing. You can actually add the vouchers under its current name, under the old name, Also, when you upload, that Gary hasn't shown that, but you can upload things in batch as a whole spreadsheet. And when you do that, you can map those names that you are uploading to the names that the Thesaurus currently accepts, or you can upload that batch spreadsheet as is, so with the old names.
Right, good question, Blanca. And thank you for the explanation.
Yeah, thanks for the question.
All right, we're past the hour, so we'll cut it off there. But thank you again. This was awesome. And Frank, I might email you for a link to your Ecuador checklist to make sure I have the right one because I want to include that in the
Absolutely, yes.
I'll send out about this. Awesome. Thanks,
It's actually
everybody. We'll see you next
manual
month.
in South America.
Okay, thanks
Sorry.
everyone.
Bye.
Yeah.